Welcome to another episode from the Veterinary Medicine Digest. Today, we're delving into a topic that is both historically significant and currently a major concern in veterinary and conservation medicine: canine distemper virus. For centuries, this pathogen has caused severe disease in dogs, and in recent decades, its impact has expanded dramatically, threatening a wide array of wildlife species across the globe. It's a story of a resilient virus, its complex relationship with its hosts, and the ongoing scientific efforts to understand and control it. So, what exactly is canine distemper virus, or CDV as it is often called? Scientifically, it's known as canine Morbillivirus, a name that places it in a notorious family of pathogens. It belongs to the genus Morbillivirus and the family Paramyxoviridae. This makes it a close relative to some well-known viruses, including the human measles virus, rinderpest virus which affects cattle, and phosid distemper virus, which impacts seals. This family relationship is more than just a taxonomic curiosity. It means these viruses share significant structural and functional similarities. The diseases they cause, though in different hosts, often follow a similar pattern of respiratory and gastrointestinal illness, followed by a profound and dangerous suppression of the immune system. Understanding one morbillivirus often gives us crucial insights into the others. The virus particle itself is an intricate piece of biological machinery. It's an enveloped virus, meaning its core is wrapped in a lipid membrane derived from the host cell. This envelope is not just a passive wrapper, it's studded with proteins that are absolutely essential for the virus's life cycle. Inside this envelope lies the virus's genetic material, a single, non-segmented strand of negative-sense ribonucleic acid, or RNA. This genome, which is about 15,690 nucleotides long, contains the blueprints for all the viral proteins. The genome is organized into 6 main genes that code for 6 structural proteins. First, there's the nucleocapsid protein, or N protein, which tightly wraps around the viral RNA to protect it and form a helical structure called the ribonucleoprotein complex. Working with the nucleocapsid are 2 transcriptase-associated proteins, the phosphoprotein, or P protein, and the large protein, or L protein, which is the viral polymerase responsible for transcribing and replicating the genome. These 3 components, the N, P, and L proteins, along with the viral RNA, form the functional core of the virus. Lining the inside of the viral envelope is the matrix protein, or M protein. It acts as a crucial bridge, connecting the inner ribonucleoprotein core to the glycoproteins embedded in the outer envelope. This connection is vital for the assembly of new virus particles before they bud off from an infected cell. And finally, protruding from the outer surface of the envelope are the 2 most critical proteins for initiating infection, the glycoproteins. These are the hemagglutinin protein, known as the H protein, and the fusion protein, or F protein. The H protein is the attachment protein. Its job is to recognize and latch onto specific receptor molecules on the surface of a host cells. Think of it as the virus's key. Once the H protein has secured a lock, the F protein takes over. It's a fusion machine that, once activated, merges the viral envelope with the host cell's membrane, allowing the viral core to be released into the cell's cytoplasm, where it can begin the process of replication. The interaction between these 2 proteins is a finely choreographed dance that is absolutely essential for the virus to be successful. In addition to these structural proteins, the P gene cleverly encodes 2 non-structural proteins, the V and C proteins, through processes like alternative reading frames and RNA editing. These proteins are not part of the virus particle itself, but they are master manipulators of the host's immune system, playing a critical role in preventing the host from mounting an effective early defense. This intricate structure and genetic makeup allows the canine distemper virus to be an incredibly effective pathogen. It's highly contagious and can spread with alarming speed through susceptible populations. Transmission occurs primarily through aerosols, tiny droplets containing the virus that are expelled from an infected animal's mouth, nose, or eyes when they cough, sneeze, or simply breathe. Direct contact with these fresh bodily secretions is another major route of infection. The virus can be found in virtually every secretion and excretion of an infected animal during the acute phase of the disease, including urine and feces. An infected animal can shed the virus and remain infectious for a long time, sometimes for as long as 90 days, even if they appear subclinically infected and show no obvious signs of illness. Like many enveloped viruses, canine distemper virus is quite fragile outside of a host. It is quickly inactivated by things like ultraviolet radiation from sunlight, heat, and drying out. At room temperature, it might only survive for a few hours in tissues or exudates. This means that transmission is largely dependent on close contact between an infected animal and a susceptible one. However, the virus can survive for several days at temperatures below freezing, especially if it's protected by organic material, which can increase its frequency during colder seasons. One of the most striking features of this virus is its incredibly broad host range. While it was first described as a disease of domestic dogs, it is now recognized as a global, multi-host pathogen. Evidence of infection has been found in a vast array of carnivore species across many families, including the Canidae family, like dogs, foxes, wolves, and coyotes, the Mustelidae family, ferrets, minks, badgers, and otters, the Procyon family, raccoons, and even large felids like lions, tigers, and leopards. There have also been concerning reports of infections in non-human primates, such as rhesus and cynomolgus macaques, which have raised questions about its potential to cross even further species barriers. A meta-analysis of numerous studies has revealed just how widespread this virus is, particularly in its primary reservoir, the domestic dog. The analysis found that, on average, a third of dogs suspected of having clinical distemper tested positive through molecular methods, and nearly half of all dogs, including apparently healthy ones, showed serological evidence of exposure, meaning they had antibodies against the virus. These numbers highlight the constant circulation of the virus in dog populations and underscore their critical role in the virus's ecology. Unvaccinated domestic dogs, especially those living in high densities in communities surrounding protected wildlife areas, are considered the main source of infection for many vulnerable wildlife populations. The interface where domestic animals and wildlife meet, at the edges of parks, in rural landscapes, or at shared resources, is a critical hotspot for cross-species transmission. Interactions at kill sites, where predators like lions and hyenas might come into contact with jackals or domestic dogs, provide a direct mechanism for the virus to jump from one species to another. This ability to infect multiple species and be maintained in a widespread domestic animal reservoir makes canine distemper virus an especially difficult disease to control and a serious threat to conservation efforts. So, what happens when the virus successfully enters a new host? The pathogenesis, or the way the disease develops, is a multi-stage process that begins in the respiratory tract and can end in the central nervous system. After being inhaled, the virus's first targets are the immune cells residing in the upper respiratory tract, such as macrophages and dendritic cells. Here, the virus begins its initial replication. This is where the first of the virus's two key receptors comes into play, a molecule called the signaling lymphocyte activation molecule, or SLAM, also known as CD150. SLAM is expressed on the surface of various immune cells, including activated T and B lymphocytes, dendritic cells, and macrophages. The viral H protein has a specific shape that allows it to bind tightly to the SLAM receptor. This binding is the critical first step that allows the virus to infect the very cells that are supposed to protect the host. Once inside these initial immune cells, the virus is transported within 24 hours to the local lymphoid tissues, such as the tonsils and respiratory lymph nodes. Here, the virus amplifies itself massively, spreading rapidly to other SLAM-positive T cells and B cells. This targeted destruction of immune cells is what leads to one of the defining features of distemper, a profound and severe immunosuppression. The number of circulating white blood cells, or leukocytes, plummets, a condition known as leukopenia. This crippling of the immune system begins very early in the infection, making the host highly susceptible to secondary bacterial and opportunistic infections, which are often the direct cause of death. In fact, experiments with genetically engineered viruses have shown that if a virus is made SLAM blind, meaning it can no longer recognize the SLAM receptor, it is unable to establish this systemic infection or cause disease, highlighting just how essential this first step is. From the local lymph nodes, the infected immune cells carry the virus into the bloodstream, a state known as cell-associated viremia, spreading it systemically throughout the body to all secondary lymphoid organs, like the spleen and thymus. Now, the virus is ready for the second stage of its invasion, which involves spreading to epithelial tissues. To do this, it utilizes its second key receptor, Nectin-4. Nectin-4 is an adhesion protein found in the junctions between epithelial cells, the cells that line the surfaces of organs like the respiratory, gastrointestinal, and urinary tracts. Infected lymphocytes circulating in the blood travel to these tissues and come into contact with the basolateral surface of the epithelial cells, where Nectin-4 is located. The virus then uses Nectin-4 to cross from the immune cells into the epithelial cell layer. Once inside the epithelial cells, the virus replicates and is then released from the apical, or outward-facing, surface of the cells. This is the crucial step for viral shedding and transmission to a new, uninfected host. Without the ability to use Nectin-4, the virus remains trapped within the immune system. Genetically engineered. Nectin-4 blind. Viruses can still cause leukopenia and immunosuppression, but they do not cause the typical clinical signs of disease, like rashes or respiratory symptoms, and they are not shed from the body. This elegant research has demonstrated a clear, sequential use of two different receptors. SLAM is the key to entering the immune system, causing immunosuppression, and spreading throughout the body, while Nectin-4 is the key to exiting the body, causing clinical signs, and ensuring transmission. The clinical outcome of a canine distemper virus infection varies dramatically and depends on a delicate balance of factors, including the virulence of the specific viral strain, the age of the animal, and most importantly, the strength of the host's immune response. In many cases, an estimated 50 to 70% of infections in domestic dogs are actually subclinical, meaning the animal's immune system successfully clears the virus without any noticeable illness. If a dog is able to mount a strong and rapid immune response, it can clear the virus from its tissues and recover completely. However, if the immune response is weak or delayed, the virus continues to replicate and spread massively, leading to severe, multisystemic disease. The incubation period can range from 1 to 4 weeks or even longer. The initial signs are often nonspecific and easily missed: a transient fever that peaks 3 to 6 days after infection, accompanied by a loss of appetite, slight depression, and a clear discharge from the eyes and nose. As the virus spreads to epithelial tissues around 10 days after infection, more severe signs begin to appear. The respiratory system is often hit hard, with symptoms like a persistent cough, difficulty breathing, dyspnea, and pneumonia. Gastrointestinal signs are also common, including vomiting and diarrhea, which can sometimes be bloody. The skin can be affected as well, with some dogs developing a rash of dermal pustules. Two of the most characteristic signs of distemper, though not always present, are hyperkeratosis and enamel hypoplasia. Hyperkeratosis is a thickening and hardening of the skin, most commonly seen on the foot pads and nose, which is why distemper was historically called hard pad disease. Enamel hypoplasia is a defect in the tooth enamel that occurs when the virus infects the developing permanent teeth of young puppies, leaving them pitted and damaged for life. While these systemic signs are serious, the most devastating and often fatal aspect of canine distemper is its impact on the central nervous system. Neurological complications can develop concurrently with the systemic illness, but often they appear later, starting around 20 days after infection, sometimes even weeks or months after the initial signs have seemingly resolved. The range of neurological signs is broad and terrifyingly progressive, reflecting the specific areas of the brain being damaged. One of the most classic neurological signs is myoclonus, which is an involuntary, rhythmic twitching of muscles or muscle groups. This is often seen in the jaw muscles, leading to the so-called chewing gum fits, but it can affect any part of the body. Other signs can include seizures, circling, head tilting, nystagmus, involuntary eye movements, ataxia, incoordination, paralysis, and profound behavioral changes or dementia. Dogs that develop neurological disease rarely survive, and those that do may be left with lifelong neurological deficits. How does the virus get into the brain, a normally well-protected site? There appear to be two primary routes of neuroinvasion. The first is the hematogenous route, where virus-infected lymphocytes travel through the bloodstream and cross the blood-brain barrier, releasing the virus into the cerebrospinal fluid. The second is the anterograde neuropathway, where the virus infects neurons in the olfactory mucosa of the nasal cavity, and then travels directly along the olfactory nerve into the brain. Once in the brain, the virus causes widespread inflammation and demyelination, the destruction of the protective myelin sheath that insulates nerve fibers. This damage disrupts the transmission of nerve signals, leading to the severe neurological signs observed. The cells that are most often infected in the brain include neurons, ependymal cells lining the ventricles, and various glial cells, which are the supportive cells of the nervous system, such as astrocytes and oligodendrocytes. This brings us to a fascinating puzzle in the pathogenesis of canine distemper: the identity of the virus's receptor in the central nervous system. Extensive studies have shown that the two known receptors, SLAM and Nectin-4, are either absent or expressed at very low levels on key brain cells like astrocytes and neurons. Yet, these are the very cells that become persistently infected and contribute to the devastating neurological damage. This paradox strongly suggests the existence of a third, as yet unidentified receptor on glial cells, which researchers have provisionally named GLIAR. The existence of this third receptor is supported by experiments showing that a recombinant virus engineered to be blind to both SLAM and Nectin-4 can still spread efficiently from cell to cell in cultures of primary astrocytes. This GLIR likely allows the virus to spread in a non-cytolytic, or non-cell-killing, manner between astrocytes, which would enable it to establish a persistent infection within the brain while hiding from the immune system. In some rare cases, dogs that survive an acute infection can develop a chronic, progressive neurological disease months or even years later, known as old dog encephalitis, or OD. This condition shares many clinical and pathological features with a rare complication of measles in humans called subacute sclerosing panencephalitis, and it appears to result from the virus persisting in the nervous tissues over a long period. Given the wide variety of clinical presentations, from subclinical infection to severe multisystemic and neurological disease, a definitive diagnosis of canine distemper cannot be made on clinical signs alone. Laboratory confirmation is essential. The gold standard for detecting an active infection is the use of molecular assays, such as reverse transcriptase polymerase chain reaction, or RT-PCR. These tests are highly sensitive and specific, capable of detecting the virus's RNA in a variety of samples, including blood, urine, or swabs from the conjunctiva, nose, or throat. More advanced versions like real-time RT-PCR can provide results even faster and quantify the amount of virus present. Another diagnostic approach is to look for viral antigens, the proteins of the virus itself. Immunofluorescence on conjunctival or nasal smears can detect these antigens, but this method is generally less sensitive and is most reliable only within the first 3 weeks of infection, when the virus is still abundant in epithelial cells. Serology, which detects antibodies against the virus in the blood, can also be useful, but its interpretation can be tricky. High antibody titers can indicate a recent or past natural infection, but they can also be the result of vaccination. One way to distinguish these is to test for a specific class of antibodies called immunoglobulin M, or IgM. IgM antibodies are typically produced early in an infection and persist for about 3 months, so their presence is a strong indicator of a recent, active infection. Beyond the immediate clinical picture, studying the genetics and evolution of canine distemper virus has provided profound insights into how viruses adapt and cross species barriers. Although canine distemper virus is considered a single serotype, meaning antibodies against one strain will generally recognize others, there is significant genetic diversity among circulating strains. This diversity is most pronounced in the gene that codes for the H protein, the viral attachment protein. The amino acid sequence of the H protein can vary by up to 10 or 11% between different strains. Based on these differences in the H gene, scientists have categorized canine distemper virus strains into several major genetic lineages, which often have a geographic association. These include lineages like America 1, which notably contains most of the commercially available vaccine strains, as well as America 2, Asia 1 and -2, European, and Arctic lineages. This variability in the H protein is not just random, it is the engine of the virus's evolution and its ability to jump to new hosts. The H protein, remember, is the key that unlocks host cells by binding to the SLAM receptor. Subtle changes in the shape of this key can allow it to fit the slightly different locks found in other species. Research has pinpointed specific amino acid substitutions in the H protein's SLAM binding region that are strongly associated with host switching events. For example, substitutions at positions 530 and 549 predominantly occur in virus strains isolated from non-canine hosts, providing compelling evidence that evolution at these sites has enabled the virus to spread to new species like large cats, seals, and raccoons. This is a beautiful example of pathogen evolution in real time, and surveillance of these key amino acid residues can help us assess the potential for the virus to emerge in new wildlife populations. A recent and particularly fascinating line of research has explored how the genetic diversity within a single virus population, known as a quasi-species, influences its ability to adapt to a new host. RNA viruses like canine distemper virus have error-prone polymerases, so when they replicate, they produce a cloud of genetically similar but not identical variants. One study compared the adaptation of 2 versions of an attenuated virus strain in ferrets. One was the original, cell culture-adapted virus stock, which had a high level of genetic diversity, and the other was a recombinant virus created from a single complementary DNA clone of that stock, which had very low genetic diversity. The results were striking. The highly diverse virus population adapted to the ferret host much more quickly and became virulent faster than the low-diversity recombinant virus. Deep sequencing revealed that the diverse population already contained beneficial mutations at very low frequencies. These pre-existing variants acted as a form of genetic memory, allowing the virus to rapidly select for the fittest genomes in the new host environment. The recombinant virus, lacking this pre-existing diversity, had to evolve these beneficial mutations from scratch, which delayed its adaptation process. This work not only illustrates the critical importance of genetic diversity for viral evolution, but also has significant implications for vaccine safety. It suggests that modern recombinant vaccines, which are derived from a single genetic clone, are inherently safer because they have erased this genetic memory, making them far less likely to revert to a virulent form. When it comes to treatment and prevention, the options for an animal already sick with distemper are unfortunately limited. As of now, there is no specific, commercially available antiviral drug to combat the virus directly. Therefore, treatment relies on intensive supportive care. This includes providing intravenous fluids to combat dehydration, nutritional support, and crucially, broad-spectrum antibiotics to fight off the secondary bacterial infections that frequently arise due to the virus-induced immunosuppression. Research into specific antivirals is ongoing. The drug ribavirin has shown some ability to inhibit the virus in laboratory settings, and newer compounds that target the viral polymerase have shown promise in experimental models, but these are not yet available for general use. Given the lack of a cure, prevention through vaccination is paramount and represents the most effective tool we have to control this devastating disease. Modified live virus vaccines, or MLVs, were first developed in the 1950s and have been instrumental in keeping the disease under control in domestic dog populations. These vaccines contain a live, attenuated version of the virus that has been weakened so that it can stimulate a robust immune response without causing disease. They are highly effective and elicit a long-lasting protective immunity that persists for at least 3 years, and likely much longer. Vaccinating puppies presents a unique challenge due to the presence of maternally derived antibodies. Puppies receive passive immunity from their mothers through colostrum, but these same protective antibodies can neutralize the vaccine virus, preventing the puppy from developing its own active immunity. To overcome this, a standard vaccination protocol involves starting a series of vaccinations when the puppy is 6 to 8 weeks old, as maternal antibodies begin to wane, and giving booster shots every 2 to 4 weeks until they are about 16 weeks old. Despite the high efficacy of vaccines, outbreaks of distemper still occur, even in vaccinated animals. These so-called vaccine failures are rarely due to the vaccine itself being ineffective. More often, they are attributable to factors like improper storage or handling of the vaccine, which can inactivate the live virus, or an incorrect vaccination schedule that fails to overcome the window of maternal antibody interference. While there is ongoing debate about whether the genetic differences between the vaccine strains, which belong to the America 1 lineage, and the diverse wild-type strains circulating globally could reduce vaccine efficacy, current evidence suggests that the existing vaccines still provide solid protection against the various lineages. The issue of vaccination becomes even more complex when we consider wildlife. The same modified live vaccines that are safe for dogs can be dangerous and even fatal for certain highly susceptible wild species, such as black-footed ferrets or European mink. For these sensitive species, safer alternatives have been developed, most notably recombinant vaccines. The canarypox vector vaccine, for example, uses a harmless bird virus as a carrier to deliver the genes for the canine distemper virus H and F proteins into the host. Since the canarypox vector cannot replicate efficiently in mammals, it is very safe but still effectively stimulates an immune response. This vaccine has been used successfully to protect a range of wildlife, including ferrets, meerkats, and even endangered giant pandas. However, the logistical challenges of vaccinating entire wild populations, from capturing animals to administering booster shots, are immense and often impractical. For this reason, a key strategy for protecting wildlife is to focus on controlling the virus in the domestic dog reservoir population through widespread vaccination campaigns, thereby reducing this spillover risk to adjacent wildlife. Finally, given the virus's demonstrated ability to jump species barriers, a crucial question arises: does canine distemper virus pose a zoonotic threat to humans? The answer, at present, is reassuringly no. There have been no known reports of canine distemper virus naturally infecting humans. A key reason for this is that the human version of the SLAM receptor is a poor match for the viral H protein, creating a significant barrier to infection. However, the story is not entirely straightforward. Concerning outbreaks of lethal distemper have been documented in non-human primates, including rhesus and cynomolgus macaques, demonstrating that the virus can indeed infect primates. Furthermore, laboratory studies have shown that while canine distemper virus cannot use the human SLAM receptor, it can efficiently use the human version of the Nectin-4 receptor to enter human epithelial cell lines. Perhaps most unnervingly, researchers have shown that a single amino acid mutation in the H protein is all it takes to allow the virus to use the human SLAM receptor in vitro. While this does not mean the virus is on the verge of jumping to humans, it does suggest that the potential for such an event, however small, exists and warrants continued vigilance. It's also been proposed that the widespread immunity to measles in the human population, either from vaccination or natural infection, might provide a degree of cross-protective immunity that helps shield us from its canine cousin. In conclusion, canine distemper virus is a remarkable and formidable pathogen. Its history is deeply intertwined with that of our canine companions, and its future is a critical issue for global wildlife conservation. We've seen how it masterfully exploits a sequence of 2, and likely 3, different cellular receptors to orchestrate a complex pathogenesis, beginning with a crippling assault on the immune system and often ending with a devastating invasion of the central nervous system. We've explored how its inherent genetic diversity acts as a powerful engine for evolution, allowing it to adapt and jump to new hosts with alarming frequency. And we've recognized that while we have a powerful tool in vaccination to protect our domestic animals, the challenges of controlling this virus at the domestic-wildlife interface remain immense. The ongoing story of canine distemper virus serves as a potent reminder of the dynamic and ever-changing nature of infectious diseases, and the critical need for continued surveillance, research, and a holistic, One Health approach to protect the health of animals and, potentially, ourselves. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging,